Hey guys, today we're going to continue with our lecture on membrane transport, and we're going to focus on active transport. So last time when we talked about simple diffusion and facilitated diffusion and osmosis, and I heard that the audio cut out a little bit on the osmosis part. So I'll go back and listen to that and see what is missing, and I'll put an extra little clip out about that. Okay, so today we're going to focus on active transport. We're going to be talking about protein pumps, endocytosis, which includes both phagocytosis and pinocytosis, and exocytosis. And we'll also talk about sodium-potassium pumps, which is the mechanism by which action potentials are propagated through the nervous system. So cool stuff. Oh, look, they move. Okay. When we talked about passive transport, we were really talking about no energy use, and we were talking about molecules moving from high concentration to low concentration. Today, in active transport, we're going to be focusing on mechanisms that use energy, which allow molecules to move against the concentration gradient. That is, from low concentration to high concentration. So, we're still on understanding number 1, but now we're going to hit the last part, active transport. So active transport requires energy in the form of ATP. And when we talk about cell respiration and photosynthesis, we'll talk a lot about what ATP is. For now, it's important that you understand that it's a form of energy currency that the cell can use, right? So when you eat food, there's glucose in that food, and we know that glucose stores energy, but glucose is like a foreign currency. Your body can't use it. Your cells can't use glucose to do things like cellular processes like active transport. So your body has to convert the energy in glucose into ATP, and that's what cell respiration is. For now, understand that we're using ATP as an energy molecule to power certain cellular processes like active transport. And because you're using this energy, you can push molecules that are at low concentrations to high concentrations. And there's 3 main types: protein pumps, endocytosis, and exocytosis. We kind of just talked about that. So let's talk about these protein pumps. Sometimes you'll see them called proton pumps if they're pushing hydrogen ions. If they're pushing other things, they might be called something else. But the word protein pumps is a good vocabulary word that you should know, because whatever they're pushing, these molecules are protein. They're composed of amino acids, right? Those amino acids, the order of these amino acids determine their shape. And in biology, your shape is your job. So these protein pumps are specific to the molecules that they push across the membrane. Now, they're embedded in the membrane and they go all the way through. So they're transmembrane, right? Meaning they stick out on both sides, which is important because their job is to get molecules from one side to the other. And like I said, they push molecules against the concentration gradient. So here you see we have a low concentration of hydrogen ions or protons over here, and we have a high concentration over here. And this protein channel's job is to push protons or hydrogen ions across the membrane to where there's a higher concentration. Each pump is specialized to transport a particular molecule, and we require energy, meaning we're going to use ATP. And when you use ATP, you don't like use it up. It doesn't disappear. You convert it to ADP, and then your body recycles it back into ATP. Okay. So when we're talking about endocytosis and exocytosis, we need to talk about vesicles, right? Vesicles are small sacs of membrane, and they move in and out of the cell. Now, it's really important that you understand that vesicles are made out of the same material that the cell membrane is, right? We have that phospholipid bilayer. We have the hydrophilic heads, and we have the hydrophobic tails on the inside. And this is to move bulk quantities of materials in and out of the cell. This is understanding number 2. The fluidity of membranes allows materials to be taken into cells by endocytosis or released by exocytosis. Okay, so really the key to this understanding is the fluidity. Membranes are not stiff. They're kind of like bubbles. They can pinch off and join, and that's exactly what a vesicle is. It's a little piece of membrane that pinches off and then rejoins later on. When we talk about the endomembrane system, we're really talking about that same membrane material forming bubbles and moving materials from the cell membrane to the Golgi and back and forth. Because the Golgi is made out of the same thing. It's still made out of that same membrane material. So when we talk about endocytosis, we're talking about bringing materials into the cell by pinching the vesicles. All right, so here we see a pit starting to form. We start to see that membrane pinch off. And then we see the vesicle. Now, this is the outside of the cell. This is the inside of the cell. This vesicle is then going to transport to the Golgi and bring whatever materials in. It might just release them in the cell, depending on what it is. Now, we know endocytosis, or you should know that endocytosis means in, because endo means in, and cytosis means anything having to do with a cell. So in the cell, endocytosis. We have different types of endocytosis depending on what's being brought in. So if we're talking about large solid particles, we're talking about phagocytosis. You're going to form that vesicle. This is often receptor mediated, meaning there are receptor proteins, glycoproteins, and glycolipids on the cell that recognize a particular particle. And then the cell brings it in using a vesicle. We have pinocytosis, which is water and liquids. Water can move across the cell membrane in a couple of different ways. We talked about how water moves through via diffusion, that's osmosis. There's also aquaporins, which are proteins that allow water to flow in rapidly. And sometimes if you need to bring in water in large quantities very quickly, you actually see that through pinocytosis as well. And then receptor-mediated specific molecules. This is kind of like phagocytosis, but it's not necessarily a large solid particle. So receptor-mediated endocytosis is when you have receptor molecules on the cell, typically glycoproteins, and they have kind of like a lock and key model, kind of like an enzyme, where they match a particular molecule. And when they receive that molecule and they recognize it, it triggers the formation of that vesicle, and then it brings it in. This is how viruses manage to hack into your cells, because they have learned, they have mimicked active sites that match the receptor-mediated molecule, so that your cell is tricked into thinking the virus is something it's not, and your cell brings it in. It's kind of like a wolf in sheep's clothing. So if we're talking about exocytosis, here we have that vesicle, right? It's going to merge with the cell membrane and push those molecules out. Okay. Here we have the movement of products from the cell out. So if you are a pancreatic cell, right, and your job is to produce hormones or enzymes that you're releasing out into the body, not hormones, enzymes, the ribosomes are going to hang out in the endoplasmic reticulum. They're going to produce those products. The Golgi is then going to package them. Here's the Golgi here. So let's start over. Endoplasmic reticulum. This is where ribosomes hang out. Ribosomes are going to produce proteins. Now, the cell is, its job is to push those proteins out to the rest of the body, right? It's like a factory. So the endoplasmic reticulum will form vesicles to send those products to the Golgi. You can see here. And then you've got that vesicle, it's going to fuse with the endoplasmic or with the Golgi body. The Golgi body is going to modify those proteins and get them ready for transport, and then put them in vesicles and then send them out. And here you see exocytosis, right? And it's pushing those proteins out. So exocytosis is the reverse process of endocytosis. Exo means exit, cell. Cytosis means cell. So exocytosis, exiting the cell. The Golgi apparatus is going to package those materials for excretion into vesicles, and the vesicles travel to the membrane and fuse to the phospholipid bilayer. They fuse to that membrane and push all that stuff out. And that releases the contents of the vesicles into the environment. So there's probably a capillary here, and that capillary is going to take up those proteins, and they're going to be transported to wherever they need to go. I don't know why I have endocytosis right there, but just in case you needed another diagram of endocytosis, it's right there. Okay, so an overview. Here we have, at the top, we have the outside of the cell. The orange is the inside of the cell. We have a vesicle made of phospholipids with the proteins inside. It's going to fuse and release those proteins out into the world of your body. And then at the bottom, we have endocytosis, which is the exact opposite. Like you could literally take this diagram and flip it, and it's the same thing. So you have a formation of that vesicle right here, right? Pinching off. And then this vesicle is free to travel to where it needs to go inside the cell. And this really all depends on the fluidity of this membrane. Now, that's why it's important to not have too much cholesterol in your diet, because cholesterol is the thing that controls membrane fluidity. So if there's too much cholesterol in this membrane, then it gets stiff. And this process of endocytosis and exocytosis is severely inhibited. So your cell can't push stuff in and out like it's supposed to. So make sure you manage your cholesterol as you get older. Okay, so here we have a nice overview of transport vesicles. So if you are a macrophage, if you're part of the immune system and your job is to like scout out bad guys in the body and eat them, you're going to use endocytosis quite a bit because you're going to ingest large particles, particularly potentially bacterial sources or foreign proteins or viruses, and you're going to destroy them, right? So you need to be able to ingest large particles via endocytosis, and you need that membrane fluidity. You need that membrane to be nice and soft so it can bud and form these vesicles. You also use vesicles to excrete waste components in the cell. Right. So this is actually a huge part of the immune system. We'll talk more about the immune system in topic 6. So macrophages, they ingest bad guys, right? They chew them up and they spit them out. And then other parts of your immune system use these chopped up pieces to teach the rest of your immune system what to look for when attacking invaders. So this process is extremely important. Neurons are another cell that use endocytosis and exocytosis quite a bit. So neurons are the types of cells that pass electrical signals through your body, and it's how your brain communicates with like every other part of your body. So these would be neurons that you would have not so much in your spinal column, but more like in your hands and in your skin and in muscle tissues and things like that. And we'll talk more about this in topic 6 as well. So you have the cell body of a neuron, you have this long axon, and you have dendrites, which are these little branchy things. Endocytosis and exocytosis are used to propagate signals across the synaptic cleft from one cell to another. There's also lots of active transport going on along the axons of these cells, and we're going to spend some time talking about that. This is application number 1, the structure and function of sodium-potassium pumps for active transport and potassium channels for facilitated diffusion in axons. Now, I like to use this the neuron is a model for all different types of transport, because we will talk about the sodium potassium pump along the axon, right? But then, like I said, there's also exocytosis and endocytosis that's used at the branches. So these particular types of cells, they're using multiple types of cell transport all the time. And that's why it's important that you need to know all of them. Okay. So what we have here is a zoomed in diagram of the surface of a neural cell, right? We have the surface of an axon. This is outside of the cell, and this is inside of the cell. Now you notice there's pluses and minuses down here, and that's indicating relative charge. Okay, and we're talking about the relative charge of the inside of the cell compared to the charge outside of the cell. You'll notice we have potassium ions, which are K, and we have sodium ions. Now, all of these ions are positive, right? We're only moving positive ions around. That's okay. If you have more positive ions on one side than the other, one side is going to be more negative than the other. So if you have more positives on the top, then here you have a relative negative charge. If you have more positives on the inside than the outside, you have a positive charge. Okay? It's all relative. Okay. So to send signals, neurons depend on the flow of these positive charges, sodium and potassium, both in and out of the axon. As the concentrations change, the signal is propagated down the membrane. And we're going to take a look at this process in detail. We have some leaky channels that allow sodium in and potassium out. And we also have active protein pumps that bring sodium out and potassium in at the same time. So we're going to watch this quick video that's going to talk about it in detail, and then we'll review.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
Okay, let's review. So the sodium-potassium pump, they send signals down the axon. 3 sodium ions are pumped out. 2 potassium ions are pumped in. The result is that you have a more positive charge outside than inside. When the charge becomes too positive, a signal is initiated that causes ion channels to open, and the signal is propagated down the line. So if we look in detail, this is a diagram of one of those voltage-gated channels that opens. So after the nerve signal is reset, or after the nerve signal is passed, the cell has to reset to its internal potassium concentration. In order to do that, those potassium channels have to open back up. And these are specialized membrane proteins. Now, remember we talked about proteins have different levels of structure. We talked about primary structure, which is the order of amino acids, right? Secondary structure, which is two-dimensional structure. Tertiary structure, which is three-dimensional structure. And then quaternary, which is when you add multiple polypeptide chains together. So when we say 4 protein subunits, that's what we're talking about. We have 4 polypeptide chains that are grouped together to form a particular structure. We have one here, kind of looks like a ball and chain. We have one here, which is like a wing that helps embed the protein in the membrane. And you see another one over here, right? So that's 1, 2, 3. And then you have the channel itself, which is 4. Remember, your shape is your job. So if there's a mutation in the DNA that codes to build this particular protein, this protein's not going to work. That's a problem. So remember, let's talk about context for a second. We just had a nerve signal pass, right? So we pumped a bunch of potassium and sodium in different directions to cause that electrical concentration gradient. Now, after that passes, the cell has to reset. Otherwise, another signal won't be able to pass through. And that's what we're talking about. After that signal passes, our potassium ion channels are going to open so that we can reset. It's composed of 4 protein subunits and a narrow pore for that potassium to flow through. These little wings are hydrophobic, which is what holds them inside the interior of the membrane. And we have the hydrophilic regions at the end of the channel, right? They stick out into the extracellular and the intracellular areas. So, potassium ion channels are voltage gated, meaning they only open when the concentration is high enough. All right, so here we have the little ball and chain that plugs it open. When there is a high enough concentration of potassium on the inside, it causes that to open, right? And then the potassium ions diffuse through. Now, when we say voltage gated, we mean when the relative voltage of the inside of the cell is more negative compared to the outside, that's what triggers this opening. And then as those potassium ions diffuse through, the voltage increases until you hit your equilibrium. So really, when we're talking about concentration gradients, we're talking about both the charge, we're talking about differences in charge from inside to outside, and that charge is controlled by the presence of positive ions, in this case, potassium. So high concentration inside, low concentration outside, right? We're moving from high to low. It's that high concentration inside that causes this channel to open, allowing these positive ions, potassium ions, to diffuse through. So this would be an example of facilitated diffusion. So here we have a diagram that shows both sodium and potassium channels in a neural cell. Now, these notes up here, the action potential stuff, that's for senior year when we're really talking about the details. But I thought this diagram would be interesting to show you how the voltage changes, right, when these ions are moving. So here we have the outside of the cell, and here we have the inside of the cell, and here we have these protein channels. You don't have to worry about knowing what the different phases of the action potential are. If you're taking psych, you'll learn about this too. But for now, I just want you to watch the change in the voltage as different molecules are moving in and out. So in the first phase, we have sodium channels opening, and sodium's going to come inside, which causes the charge inside the cell to increase. And then the second phase is we have the potassium ion channels open, they go out, and that brings the charge back down. So positive in causes more positive inside, positive out, less positive. And then there's a third part where everything's like resetting a little bit, so we'll watch it. Undershoot. And then we come back. Okay. This is another way to look at it. So when we talk about the signal being propagated down the axon, you can see that these channels open and close in a wave, which causes that signal to transmit down the axon. Now, this is important because when we talk about toxins and poisons that paralyze, this is the impact that they're functioning. They're either impacting. Did I just say this is the impact that they're functioning? I did say that. This is the function that they're impacting. So they're either interfering with the ability of these channels to open and close so that the signal isn't transmitted down the axon, or they're interfering at the synapse where the signal is transmitted from one nerve to the next. And we'll spend more time talking about that senior year too. It's actually pretty cool. Okay, so at this point, you should be able to answer these questions, right? Define diffusion, that's passive, outline the process of simple diffusion, outline the process of facilitated diffusion, compare and contrast molecules that are transported through simple and facilitated diffusion, define osmosis, define hypotonic, isotonic, and hypertonic, outline the direction of the water molecule movement during osmosis, and discuss the importance of using isotonic solutions with tissue samples. You should also be, that was all the passive stuff. Now, for active stuff, you should be able to define active transport, outline the function of protein pumps, outline the process of endocytosis and exocytosis, and describe how vesicles can be used to transport materials within the cell. So I had initially planned on you creating a model of all this today, but as I was looking at the simulations for passive transport, I noticed quite a few of them are not finished yet. So what I would like for you to do is I would like for you to finish those simulations today in class. I know the due date is the 11th, which is Friday, but I'd really like you to get those done before we start our osmolarity lab. So after this lecture today, I would like you to work on that in class, and then I'll figure out what I want to do about the models in a bit. So have fun. If you need anything, let me know. Today you're taking your notes and finishing section 1.4. Make sure at the end of your notes you can answer these 13 questions. That'll help you get ready for a quiz. And then once you're done with that, I'd like you to finish off the passive transport simulations before we move any further. Okay? All right. Thanks, guys. Talk to you soon.